Thank you for the introduction. So I'm going to talk about using human pluripotent stem cell derived dopaminergic neurons for Parkinson's disease. The work I'm going to present here coming from my academic lab located at the Buck Institute in the San Francisco Bay Area. And part of the work also coming from Excel Science, which is a company I set up, leased the space here at the Buck Institute. So let me just start with a very brief introduction of the disease. Parkinson's disease is a neurodegenerative disease involving motor function caused by the death of dopaminergic neurons in the substantia nigra. So currently there are many treatments, for example, like increased dopamine levels in remaining cells, or give a pseudodopamine like the dopamine receptor agonist, or preventing breaking down of dopamine in the brain. There are also surgical procedures available, for example, like deep brain stimulation. But all these are just treatments. There is no cure for the disease. So this leads to what I'm going to talk about, cell therapy. In the past 3 decades, there have been clinical trials using fetal cells, so pioneered by the Swedish group and the US groups. So those studies really proved the concept of using cell as cell replacement therapy for Parkinson's disease. For example, the graft dopamine neurons, they can reinnervate the striatum. They can restore regulated dopamine release and also improve the motor function of the patient. But there are also a lot of problems associated with fetal transplantation. One issue is the limited materials. For example, one patient, you have to use about 4 to 6 fetals and combine the cells. And of course, when you are using such cell prep, you have a lot of variabilities of the outcome. And the worst side effect is the dyskinesia. So we think all these issues could be addressed by using alternative cell source, which is the focus of my presentation in the next 10 minutes. We're using human pluripotent stem cells as the start point. And also, not only these cells can be obtained in a large numbers, they, in theory, also, because of the IPS technology, so in theory, you can use autologous therapy. So this is really what I've been focused in the past decade when I started working at the National Institute of Health using human embryonic stem cells to derive dopamine neurons. So what I want to tell you a bit about is our recent progress in terms of getting an IND with the FDA. So what does an IND process entail? It's really start with academic idea with lab validation, of course, followed by the manufacturer. And this manufacturer need to meet the requirement of the regulatory. In the US, it's the FDA, and someone has to perform preclinical and clinic studies. So here is the strategy, what we have done. So first, we need to obtain clinic-grade cells, and in this case, we talk about both iPS and EA cells, and manufacture dopamine neurons in the cGMP facility, evaluate efficacy, safety, and the requirement for immunosuppression in suitable animal models, and design clinic protocols. So at this stage, we are in the progress of finishing up this and assemble the IND. So let me just start walk you through the process. So first, we have developed a stage-wise manufacture process of manufacture dopamine neurons, and start with either embryonic stem cells or iPS cells, and then we make an intermediate stage called the neural stem cells, which can be cryopreserved and also further differentiate into neurons and glials. And differentiate them further to dopamine precursor, this is the stage where we cryopreserve it and then transplant. Okay, so as far as I have known, this is the first report that we use the cryopreserved cells for clinic use and directly. We will freeze the cells down without further on-site manufacture to inject it into the patient. And these are the in vitro step, which really just to test if the cells can be mature in vitro. This is related to the potency assay. So this process has been transferred successfully to a CGMP facility. In my case, this is the City of Hope CGMP facility. So we have in the past 3 years and under a grant from California Institute for Regenerative Medicine, we have run 3 dry runs. And these are the cells we have manufactured. As you can see, is in a large number, which is enough for all the preclinical studies, and is intended for a phase 1 trial. But this is the dry runs. It's manufactured in the CGMP facility, okay? And we have used these cells to perform some preclinical studies, including efficacy and safety studies, as well as the surgical device test. I'm going to show you just a couple of these results. So for the efficacy, we use the 6-hydroxy dopamine lesion rat model. So this is a well-established model, lesion one side of the striatum and then transplant. So these are the group of animals receive the control, and we also have a better control. This is the cryopreserved cells 4 days before the final product. This cells is considered immature and may not provide functional improvement. And this is the product stage, day 14 cells. At the time when we start this experiment, and since it's a 9-month study, I cannot really take any risk if the cryopreserved cells didn't work. So we also have a side-by-side study using the fresh reculture cells. So these are the behavior outcomes. So after 3 months that you can see the motor function is significantly improved, and this is done by the amphetamine-induced rotation test, which you can see the improvement increase and persist at the end of the study, which is 6 months. And we don't see any difference when using freshly prepared culture or use the cryopreserved culture. So both the control group didn't provide any functional improvement. So the function improvement is really associated with the survive and the differentiation of dopaminergic neurons. This is the graph, histologic analyzed at the graph side, which you can see after 4 months of transplantation. Look at the day 10 control. You do see human cells, which is labeled by stem 1-to-1, but you don't see any dopaminergic neurons here. This is an enlarged mag of the same photo, and see the freshly cultured cells that you can see a lot of dopaminergic neurons survive and graph here. And the same goes for the cryopreserved cells, which you can see. Now, this is just another stain with the human nuclear antigen together with TH. Again, you see the control group. You see the human cells, but you don't see the dopaminergic neurons, whereas you see the clear survive and in both the fresh reculture and the cryopreserved cell receiving group. So these are additional analyses with the midbrain markers, for example, like FOXA2 and LMX1A. This is just shown with the freshly prepared cells and the cryopreserved cell transplant group. Again, you can see the fine axon extension of the Th positive cells, which remain expressing the midbrain markers. So the other study I want to tell you a bit about is the surgical device. As we are preparing for the IND filing, so we chose to use a FDA-approved stereotactic platform, which is already used for deep brain stimulation. The device we actually choose is one of what we are going to choose. We have done some tests. This is a radically branch deployment system. This is developed by my collaborator Daniel Lim at UCSF, and this is again the cassette attached to the stereotactic platform. And using this device, and Daniel has done the transplantation under the real-time MRI guide with these cells labeled, and you can see actually when you deploy the cells, you can see 1, 2, 3, 4, there are 6 of them. Okay, and this is done in mice. Actually, it's very difficult to do the surgery. So when we talk to the FDA, ask about the device, and they indicate they want to see the safety data from a large animal model. So that's where we go with a swine model. Again, this is our cells as exactly the developmental stage, day 14 cells, which I show you the efficacy data, and we use a label line. And in this case, we have the GAP label, and we injected this with exactly the same device I just showed you into the striatum of the swine, the pigs here. And this is 2 weeks after injection, which you can see the human cells both labeled by the GAP and also confirmed by human marker STEM121. So the paper has been published in Molecular Device earlier this year. So I'm not going to show you the data, but I do want to tell you we have done the tumorigenicity study, since this is a pluripotent stem cell-based therapy. So this is a 9-month study and done with the Jackson lab, usually a mouse, and looking primarily at any possibilities of tumor formation, which we didn't see any. And we are also at the same time doing the tox studies. So I'm going to end with, these are all the data I've been showing you from the human embryonic stem cell line H14, which has been manufactured by Weissman Manufacturer at Wisconsin. One of the lot funded by the NIH, which we have been using for deriving dopamine. So I'm going to walk you through, just show you 2 slides, the iPS cells we have been using. So this is our strategy to obtain cGMP iPS cells product, which basically start with tissue banking. We decide to use the CD34 positive cells as the starting material, and then get to the banking derived iPS cells, and go further to make neural stem cells expansion, or may need the selection and transplantation. This is the workflow. So I'm going to ask you not to take the picture of the next 2 slides. Okay, so these are still works undergoing, please. This is the lines that has been manufactured right now at Lonza. Okay, this is as far as my knowledge, is the first real cGMP iPS line. This is manufactured by Lonza under an NIH contract. Okay, so what we have shown here is you can start with CD34 positive cells with nucleofaction and make iPS cells, and go through all the characterization, ability to differentiation, and of course normal carrier type and the DNA fingerprint analyze. Okay, and this is the line I have acquired, and we study the neural differentiation with this line together with Lonza, and this is how we make rosette and make neural stem cells. Okay. And also I want to show you just one more face picture, or the picture images of we have been able to engineering it. This is by genome editing. And in this case, we use the GAP just to target at the safe harbor. So in case we need the label cells for preclinical studies, and also for later for genetic engineering. Okay, so I'm hoping we are submitting this paper this week. So hopefully, after the study, and this will be the line with the intention that will be make it available to the research community. So with this, I'm just going to summarize where we are on the PD therapy. So we had pre-IND discussions with the FDA. We have access to clinical-grade EAC and IPS line, as well as the matched research grade cells. We have identified the manufacturer site and access to the drug master file, and we have performed preclinical studies with the cells manufactured by the cGMP compatible process. And we have developed engineering subclones of the IPS line together with the H14 lines too. We have coordinated with clinicians on surgical trial design. And these are the papers we have published to support this IND enabling effort. So we have the manufacture process, preclinical studies, device test, and the engineering approach. So with that, I want to thank the people who contribute to this work, and my lab at the Buck Institute, and people at the Excel Science, and my many collaborators. This is just three of the few. Mahendra Rao was the founding director at National Institute for Center for Regenerative Medicine. So he quit there last year, and he has worked with this effort with me and my long-term collaborator Daniel Lim, working with the device, and also this work is really the cGMP IPS line is done at Lanza. So my collaborator, Tom Sperner, there is in charge of this project. So I'm not sure I have time for questions.
I must have scared
you. Just one rapidly. I'm just curious, how do you are accreditation for GMP, good manufactured product?
How do I?
Yes, do you have an inspector that came on the seat? Do you have a standard? Or is your company that decide that you are GMP level?
I don't define that. This is a FDA-approved inspector cGMP manufacturer of Lanza located in Waxwell in Maryland. So I don't define, but everything is followed from the donor consent and acquisition, and every line they have like hundreds of pages you have to follow.